Now here you can see in today's class we would be dealing with fertilization in flowering plant. So fertilization basically it is the fusion of the male and the female gamete, but see, fusion on the male and the female gamete is termed as syngamy. Now what is syngamy? The syngamy means it should be between compatible species. Intercompatible means species of the same type. Now basically in general theory of fertilization, one gamete is motile, that means capable of movement, and the another one is sessile, that is not capable of movement. So basically that what is motile is smaller in size, and that what is sessile is bigger in size, and the sessile one later on develops into embryo, or rather into the new offspring. See, now the male gamete here in the flowering plant is termed as pollen, and the female gamete here is termed as ova, and basically ova is sessile. Now if we recollect the study material we had discussed earlier, the basic diagram of a female, that is known as a pistil. That pistil begins with the stigma, style and the ovary, and that stigma, style and the ovary remains sessile, and what is motile is the male gamete or the anther, that is known as germ pore or pollen grain. Now here see, there are certain steps that I have classified stepwise. Now the first one is the pollen pistil interaction. Now what is pollen pistil interaction? First, basically this pollen pistil interaction is identification of genetically identical pollen. Because see, every flower has got a separate stigma, a separate genetical material and a separate pollen. For example, if hibiscus flower has to be fertilized, the pollen has to come from the hibiscus because genetically they are similar. So it is here, since the ovary has no connection with the stigma or directly the pollen cell, the identification is done here itself, and that is basically known as the pollen pistil interaction. Now here you can see a small one, I have marked it as a germ pore. Basically it is a single pollen, and this single pollen I have drawn for your convenience, but if you can recollect the slides that we have I've shown you, the slides shows that this stigma can get attached to many pollen grains, and many pollen grain can germinate together, but only one single pollen grain has to be fused with the ovule. Okay, now here see, while the pollen tube grows inside the stigma, while the pollen tube grows inside the stigma, there is a certain secretion from the stigma that forms the base of the attachment of the pollen grain with the stigma. So what is the secretion? The secretion is basically water and sugar, and this secretion actually forms the base of attachment of the anther and the it's of the pollen grain and the stigma. Now here you can see in the second diagram that the pollen has got a small opening. See, if you can see a black dot I have marked, and that is known as germ pore. What is germ pore? A small opening in the pollen grain, and you see the outside, these are known as the exine. The exine is very tough, and the pollen tube cannot attach or germinate from the exine. So a small opening, that is known as a germ pore, is there, and through that germ pore a small pollen tube develops. Now you see the germination, and the third stage here is the germination of the pollen, and it is through the germ pore emerges. Okay, now during this germ pore emerges, you see the fourth point. It is written the growth of the pollen tube. Obviously, when the growth of the pollen tube starts, it is in the two-cell stage. What is two-cell stage? One bigger tube cell, and the second is a smaller generative cell. Now at the tube, what is the function of the tube cell? Tube cell helps in the penetration of the pollen tube, right through the style. Now you see here again that second Here you have see I have marked 3 cells. First it was 2 and this second generative cell. Second generative cell, it divides into 2. You see the second smaller generative cell undergoes 1 mitotic division and 2 male gametes are formed. Now here you see after the male gamete is formed, here it's a much broader diagram. You can see these 2 male gametes and the tube cells which later degenerates. Now here I have seen, you can see I have marked this line diagram and it is known as callus plug. What is callus plug? Callus plugs actually prevent the genetic material from going back into the tube and it also guides the nucleus downward. Prevents, here it's written, it prevents backward flow and keeps the nucleus at the tip, because the nucleus at the tip is going to fuse with the egg cell to form the zygote. Now you see, if you are seeing this diagram, now this is basically a diagram of a common angiosperm, but here has got the entry of the pollen up to the ova. See here, if you can recollect the previous thing, we are here. Now you see the pollen enters through the chalazial end, that it is known as chalazogamy. If the pollen tube enters through the integument, it is known as mesogamy. And if the pollen tube enters through the micropylar end, we call it as porogamy. So this porogamy basically is a very common method which we would be following. Now here in our earlier classes, we had told you that this is basically an anatropous one. What is anatropous one? It is inverted one. And here this diagram has been enlarged here. Now this is basically one big egg cell. Now this big egg cell has got 3 cells, that is at the antipodal end. Center 2 cells, you can see this is known as the polar cells, and here is the main egg cell or the ova and 2 synergids. Okay, now from this here we have got a micropyle end and another is known as a filiform apparatus. So what is filiform apparatus? Filiform apparatus actually guides the entry of the nucleus and to fuse with this cell. Now here you can see this synergid, we have 2 synergids. One of the synergid actually degenerates for the entry of the nucleus, and here the pollen tube actually ruptures and releases the 2 male nucleus inside, and out of the male nucleus only one fuses with this ova. Now that if it fuses with the ova, it forms the zygote. Now here you see again, it is that fertilization, the anther cell and the egg cell fuses together from the zygote. The anther cell is haploid, the egg cell is also haploid, but the diploid is the zygote. Now in the second case, the 2 male gametes, the second male gamete fuses, see 2 male cells were there. So one fuses with the egg cell to from the zygote and the other fuses with the 2 polar nucleus. You see here the 2 polar nucleus is there. The second one fuses with these 2. Okay. So when the polar nucleus fuses with these 2, so they are also haploid. So here they form a triploid. So this step is known as a triploid fusion, and basically these 3 nucleus, the triploid, forms the primary endosperm layer. Okay. Now here you see zygote develops into the embryo. The primary endosperm develops into the endosperm. Now what is endosperm? Endosperm is actually the nutritive tissue. What is nutritive tissue? That gives nutrition to the growing embryo. So that is why fertilization in plants is known as double fertilization. Why double fertilization? First the fertilization between the egg cell and the male cells, and second is the triple fusion. This is basically the process. Now here you see this method was explained by Novartism and we follow this process. Okay, so